Hello, I'm Andrew and currently undertaking my fourth year and master's project in the School of Natural Sciences at Bangor University, Wales. This poster illustrates proposed research investigating the relationship between cerebral circulation and animal cognition across the taxonomic classes Mammalia and Aves. It is widely accepted that the brain is a metabolically expensive organ to develop, evolve and maintain. This expensive brain in humans is supplied by two pairs of arteries, the vertebral and internal carotid. The vertebral arteries pass through the transverse foramina in the cervical vertebrae before joining to form the basilar artery, which, with the two internal carotid arteries, forms the cerebral arterial circle of Willis in the brain. This posterior-anterior connection allows for a large total volume of blood to circulate throughout, therefore permitting for the maintenance, evolution and expansion of a more complex neocortex. A landmark study by Baldwin and Bell in 1963, investigating sheep and oxen blood supply, showed that these animals, who are classically seen as less intelligent, had a circle of Willis that only consisted of the internal carotid arteries, which supplied the majority of the brain. The vertebral arteries did not form the basilar and only supplied the posterior section. Furthermore, they also found that there was very little mixing of blood between the internal carotid arteries and vertebral arteries, suggesting a separate anterior-posterior supply, unlike that seen in humans. Subsequently, the total amount of cerebral circulation and supply was lower. If there is less total cerebral blood flow, then it is possible that the brain will not develop to incorporate the higher information processing capacity that is observed in the highly energetically demanding human brain. If higher brain complexity in the form of neuron count, surface area, conduction velocity and gyrification arise in part as a product of large total cerebral volume, then blood flow may be correlated with higher cognitive abilities and thus some form of intelligence. This study aims to analyse diameters of the transverse foramina in cervical vertebrae to assess the vertebral artery blood supply to the brain and its relationship with animal cognition and behaviours indicative of higher order cognitive ability across the taxonomic classes Mammalia and Aves. This will be undertaken through geometric morphometrics on vertebrae C1 to C7 with measurements of the transverse foramina, which are an accurate indicator of vertebral artery diameter. Geometric morphometrics will also be undertaken on the vertebral body and canal to adjust for size across the data. Key landmarks that will be used have been identified here on Figure 1. Vertebral artery blood flow volume will be calculated from a size-adjusted transverse foramina measurement using adaptations of internal carotid artery blood flow volume studies and derivations of the shear stress equation as shown in C. Moore et al. 2019. A literature review of factors that are indicative of an animal intelligence, such as tool manipulation, highly complex social interactions or self-recognition, will be inputted into a database and ranked in a binary system for evidence of presence or absence. No evidence of the factor being present will result in an absent mark, and the total per species will be calculated and assigned to that species for data analysis. Measurements of cranial height, length, circumference and width will be used to calculate relative brain size and the encephalization quotient, which, when used with animal cognition literature, will form a basis for quantifying higher order brain functions. Finally, for statistical analysis, a single linear regression of vertebral artery blood flow against relative brain size, the encephalization quotient, and total assigned species rank across both taxonomic classes will be undertaken to allow directly comparable results between Mammalia and Aves. The first prediction of this study is that there will be a relationship between vertebral blood flow to the brain and animal cognition. Secondly, that animals who are typically described as more intelligent, such as primates, cetaceans and corvids, will all demonstrate larger blood flow volumes and subsequently larger relative transverse foramina measurements than those thought of as less intelligent, such as sheep and bovines. The relationship between cerebral circulation and animal intelligence is poorly studied, but this project will shed new light on animal cognition. I would like to thank my supervisors for all their support and guidance, and the Anatomical Society for allowing me to present this poster at their Winter Virtual Meeting 2020. Thank you for listening, and if you have any questions, please do not hesitate to get in touch with me. Thank you.